All right. Thank you very much. It's great to be here. I think a year ago, this would have been quite a strange thing to be talking about, I think, at a Dairy at Guelph research showcase, but I'm delighted to be here today, and hopefully I'll be able to give you some information that'll at least be interesting for you. Okay. So this is sort of where we're at, the detection of avian influenza in dairy cattle in March of 2024 has made this topic relevant to you all, I think, to at least some degree. And I just want to mention this is the first time that these avian influenza viruses had been detected in cattle. And it was really a surprise to everyone that this took place, that this transmission event took place. So what I want to do today, I'm going to give you a little bit of background information about the emergence and spread of avian influenza in North America, focusing on Canada and Ontario. I'll talk about the kind of current situation where we are today, and I'll describe our surveillance, how we do our surveillance in wild animals, and how that's changed over the sort of course of this current outbreak. I'm also going to describe, hopefully, why this is important work, not only for wildlife health, but also for domestic animal health and human health as well. And of course, there will be time at the end for any questions. Okay, I'm just going to give you a little bit of background information, not too much, don't worry. I just wanted to make sure we're all on the same page here. So avian influenza, this avian influenza is caused by infection with an avian influenza A virus. There are other influenza, there's B, C, and D type viruses, but we're focusing on these avian influenza A viruses. These influenza A viruses are divided into subtypes based on proteins on the surface of the virus. And you just need to know that you'll just hear these terms. So there's these two proteins, hemagglutinin, which is represented by an H, and neuraminidase represented by an N. And this current outbreak that we're dealing with right now, you may read in the news, it's H5N1, okay? Wild aquatic birds are the natural host for almost all of the avian influenza virus subtypes that we see. And the other thing that's important to note that you'll hear about in the news is that this low pathogenic and highly pathogenic. And all that means is that it's based on molecular characterization of the virus, but also the virus's ability to cause disease and mortality in chickens. We have low pathogenic that don't cause disease in chickens, and highly pathogenic when we see disease in chickens, okay? So historically, this is the situation that we were dealing with. We had low pathogenic strains of avian influenza in wild birds. Occasionally, these low path strains would get into poultry facilities. On these poultry facilities, these viruses could mutate. So these are RNA viruses. They make lots of mistakes when they replicate, and in some cases, those low pathogenic viruses turned into highly pathogenic viruses. So all that means, they became able to cause illness and death on poultry farms. And this was devastating for the poultry farms when this happened. It caused high mortality. It could spread among barns and then to different farms. But really the role of wild birds is as a reservoir. At this stage, they weren't getting sick. They were just a reservoir of all the different viruses. All right. So we have been doing surveillance for avian influenza for since 2005. There's been this sort of concerted effort. It's a collaborative effort among federal, provincial, territorial government agencies and non-governmental organizations and academic institutions. And because of the situation at this time, we had two main goals. We wanted to expand our understanding of avian influenza viruses circulating in wild birds. We wanted to understand what low-path viruses were out there. And we wanted to do that because we wanted to enhance our ability to detect highly pathogenic avian influenza strains. And we were really on the lookout for these H5s and H7s. Those were the ones that were most likely to mutate and change into highly pathogenic strains in the poultry farms. Okay, so that's what we were doing up until the emergence of this new strain of avian influenza, new for us anyway, strain of avian influenza in 2021. It changed things considerably. So this particular virus is known as highly pathogenic avian influenza. It's clade, you might see this again in the news, clade 2344B, and it's really made a big difference. It emerged a number of years ago in Asia, spread to Europe, and then in the fall of 2021, there was this emergence event, spread to North America on the Atlantic coast, and you'll see there was another incursion that's been noted here in February 2022. And all these movements from Asia to Europe and then across North America have been associated with the movement of wild birds. Now, when we think about wild birds, we don't think about them. They don't typically migrate across the Atlantic, but there is a meeting point for birds up here in Iceland. So we think probably what happened is that an infected bird from Europe in Iceland was mixing with a North American bird that was not infected. That North American bird made it back to the Atlantic coast. And this map shows the situation as of January 2022, just a few detections along the Atlantic coast of North America at that time. So what happened then? We typically think about migratory birds, which are the reservoirs of this, moving north-south. So how did it spread across? Well, you can also see here there's a lot of mixing of birds. Even though we think of these are flyways that the birds use to move during their migration periods, but there's enough mixing, and this virus was able to spread across North America in a relatively short period of time. Okay, and this is the almost global distribution of avian influenza since October 2023. So these are just cases that have been detected since October 2023. And you can see this amazing distribution of this particular virus, this H5N1 high-path avian influenza virus. And this is the one that we're really focused and we're going to talk about today. But I did just want to bring your attention, there's all these other avian influenza viruses circulating as well and being detected, and we need to keep an eye on those viruses as well. But our main goal, and particularly for this talk, is to really discuss this H5N1, which is the dominant strain globally at this time. Okay. So this strain of highly pathogenic avian influenza virus was very different than what we had seen before. You know how I mentioned that we had cases in birds, but that they weren't in wild birds, but they weren't dying, right? Now we see mortality events. So these are dead birds on a marsh. This is typical of what we have seen in some situations with avian influenza. And that is very unusual and was a new situation for us to deal with in terms of wildlife surveillance. So it changed and forced our objectives for our surveillance program to expand at this time in recognition that wild birds were not just reservoirs, but also victims of this virus. And many wild birds were dying at this time. So we needed to identify the presence, distribution, and spread of this virus and other avian influenza viruses, identify wild bird mortality and morbidity, so sickness and death associated with high-path AI, and then identify the range of species that were infected, and then make sure that the viruses that were detected were characterized, because that's really important to understand the spread. Okay, so this surveillance has been done across Canada, and I'm just going to describe how it works, just for your information. So in Ontario, the Canadian Wildlife Health Cooperative, which is housed at the Ontario Veterinary College at the University of Guelph, it is the primary point of contact for people who find dead wildlife in the province. We rely on opportunistic submissions of dead wildlife, and most of our submissions come from wildlife rehabilitators, the public, or government agencies. And this picture here, this is a dead Canada goose. They were commonly found dead, and it's caused by and the mortality was caused by avian influenza. They tend to be in quite good body condition, and it appears that they likely died acutely. So it's a sudden onset and death is what we typically see. If we see them before they die, they often have neurological signs. So this particular virus in these birds is causing neurological signs, so it's going to the nervous system and affecting the brain. So it's a nasty, nasty virus in these species. Okay, so how did we get these birds in? People can phone us at our regional center or report cases online. We'll get in touch with you and we'll try to get the carcass in for testing. The cases that come to us, when a bird is submitted to us, we collect these oropharyngeal swabs. You can see that's being done here. We'll also collect huecal swabs. And at the same time, we'll also do a postmortem. So the CWC, we do the avian influenza surveillance, but we also take carcasses in, any well-dead animal, and we try to determine cause of death. And this is our wildlife pathologist, Dr. Ryan Stevens, starting his necropsy on this gull. And we do this to try to figure out why wild animals have died, and that becomes relevant a little bit later on. That component of the surveillance becomes relevant a bit later on. Okay, so in addition to the dead bird surveillance, the Ontario Ministry of Natural Resources and Environment and Climate Change Canada also do live bird surveillance. And this is done on healthy birds to try to better understand that reservoir component for this new high-path strain. So here, this is airboat sampling. It's done at night. They go and scoop the birds up in nets. And so this is a technician. You can see the little duck there getting an oral swab done. And that animal will be released. It's a healthy animal. It'll be healthy bird, and it'll be put back in the environment. Regardless of how the samples are collected, they are submitted to a provincial diagnostic lab, which for us in Ontario is the Animal Health Lab at the University of Guelph. Any positive or inconclusive samples are then sent to the National Centre for Foreign Animal Disease, which is part of the CFIA, or is housed by the CFIA in Winnipeg, and that's for confirmation and further identification and characterization of the virus. And then this information is shared by CFIA with the World Organization for Animal Health, and that's required because this is a reportable disease. So all the positive cases in wildlife will be reported to the World Organization for Animal Health. Okay, so the information is also available to the public, and I just wanted you to be aware of this. You can just Google it, but also if you're interested, you can follow this link, and there's a dashboard available on high pathogenicity, avian influenza, and wild lice, and it has reports from across Canada of all the positive birds that have tested positive and other animals that have tested positive for avian influenza. And you can actually go in this dashboard. You can look specifically for Ontario. You can look within the last month. You can see the temporal patterns and positivity over time. So if you're interested in influenza, this is a great resource. But I did want to share some of the data and outcomes of the wildlife surveillance that we do at CWC, and just in a little more detail. And so this map shows all the submissions that we've had in 2023. The yellow dots here are the negative cases, and the red are the positive cases. You can see we have pretty good distribution pretty good submissions from Southern Ontario, but there are a lot of gaps still in our surveillance. The live bird surveillance helps to fill some of these gaps, some of the spatial gaps, and in addition also, of course, lets us know which birds are dying from the virus, which we get through the dead bird surveillance, and which ones may be acting as reservoirs, so testing positive but healthy. So that lets us get a better understanding of the whole transmission pattern of the virus in birds. Okay, and I know people are quite interested in which species are affected, and so these are the sort of overall numbers by taxa, kind of groups of birds that are of interest. So you can see sort of waterfowl, water birds, shorebirds, raptors, and corvids are kind of the main groups that are testing positive in Ontario. Some are probably around 20, just under 20% of those that come in are positive. And we haven't, at least at this time, detected any songbirds, upland game birds, pigeons, or doves, or any of the other birds. We just haven't detected it here in Ontario. But, so even though we haven't seen high-path AI in pigeons or grackles or starlings, these species have tested positive in other areas, so it doesn't mean they can't be infected, it's just that we haven't seen it yet. But it clearly isn't very common. Okay, so we've tested probably over 3,000 animals since 2021, and this figure shows a temporal distribution of animals that have been tested over time since the start, well, not since the start, but since the beginning of 2021. You can see kind of fewer samples here, and then as we started getting more and more concerned, you can see the samples increase. So this is a stacked bar graph, so if this is the total number of submissions. You can, let's say for this example, somewhere around 170, maybe 110 of them are negative, the blue, and then on top of that we have about, let's say 60 positive samples. So the orange represent the positive cases. And you can see there's a lot of fluctuation, both in terms of the numbers of samples that are submitted, the numbers that are positive. And these These changes are likely impacted by a variety of host and environmental factors and the opportunistic nature of the surveillance. Remember, we have to wait for people to call and let us know that they found a dead animal on the landscape. So one thing I wanted to point out just quickly is that the last case that we had in Ontario in dead wild animals was in, I think that's April or March of 2024. We haven't, even though we're testing a fair number of samples, we haven't had any positive cases since then in the dead birds. But the live bird surveillance, they're still detecting at low levels. There's still avian influenza on the landscape. So just because we haven't received any carcasses or animals dying of it doesn't mean it's not out there. So just be aware of that. And then the other thing I just wanted to point out is this case here in March 2022. This was the very first case of high-path AI in North America. Sorry, not in North America, in Ontario. And it was in a red-tailed hawk, like this species here. And we were able to share this information, obviously, with OMAFA and obviously with CFIA, and they were able to use this as an early warning for poultry producers. So there was, information put out. We detected this case about a week before the first poultry outbreak. So wildlife surveillance is potentially useful in that way as kind of an early warning. It did seem to work out in this case. All right. So this slide shows, this is a blog article just off the internet from the CWHC talking about avian influenza detected in wild foxes in Ontario. We knew that avian influenza could infect wild mammals, but they weren't part of, through experimental trials, and also there's been cases of wild mammals being infected in Europe, but they weren't part of our regular surveillance program. But because we do that postmortem testing that I told you about, we were able to detect avian influenza in these foxes. So the foxes had neurological symptoms, they died, we were able to do testing and examination. Samples were submitted for avian influenza and they tested positive. And these were the very first cases in North America. And I think we've all heard in the news about subsequent cases after that. So we've probably tested about 484 mammals as part of our surveillance, probably about 24 different species. We've had about 35 mammals test positive to date, including skunks, raccoons, feral cats, a mink, fisher, and an otter, and foxes. And these mammals, we think, are becoming infected after exposure to infected wild birds. So there doesn't appear to be any mammal-to-mammal transmissions, at least at this time in Ontario. They all seem to be sporadic cases resulting from contact with infected and dead birds. Okay. So I hope it's clear that this virus that we're dealing with right now is really different than other avian influenza viruses that we were dealing with in the past. Not only does it infect wild waterfowl, it also infects and causes disease in other wild birds, including raptors and other scavenging birds. And we've seen sporadic cases in wild and domestic mammals. So I put a picture of a cat there, but there's also one report of avian influenza in a dog in Ontario. So we haven't detected, the royal we, there have been no detections of avian influenza in cattle or other livestock in Canada yet, and there have been no domestically acquired human cases of this particular high-path strain of avian influenza in Canada. But the situation in the U.S. is different, and I think we really need to keep a close eye on that. So there are reports in the U.S. of positive alpacas, or a positive alpaca, a goat, and most recently a pig, and of course there's the ongoing outbreak in dairy cattle that we need to keep an eye on. And there's about, when I looked this morning, I think there's been 44 reported human cases, and they have been associated with both poultry and cattle exposure. So this is an ongoing issue and something that we should be watching out for. The other thing, so I'm just going to touch on this, on the cattle situation here. There has been in the US that these cattle cases are associated with mammal-to-mammal transmission, and this mammal-to-mammal transmission raises a lot of concerns because there's worry that the virus can adapt and become better able to spread amongst mammals and potentially to people, and then that raises a lot of concerns about increased risk for human infections, and which we want to avoid. So So that's sort of shown here. So this is, so we've got infected cattle. They were able to infect these peridomestic mammals, including cats and other wild mammals on farms. It looks like the cattle also went back and infected some of avian species. And then there's been spread amongst cattle to humans, and then spread between farms through movement of cattle. Okay? And that's just in the US, and that's our experience to date. What I want to point out to you, the other interesting thing I think about this diagram is back here. How did the cattle become infected? So if you look, this is the current idea now, is that this particular genotype that cattle are infected with, this B3.13, which you've probably heard about, likely originated from two different strains that were in wild birds. They came into this unknown host, and in this unknown host, so it was a co-infection, and parts of two of these viruses, both of those viruses sort of recombined to form this genotype 313. And then that spread to some other wildlife species, but then there's this mystery unknown host of how this virus actually made it into cattle. So, you know, it's interesting. There's still a lot of unknowns, and I guess from my perspective, it sort of highlights the, you know, there's still a lot we don't know about this virus, but I think we need to really be using this One Health approach, which we just sort of describing up here. We are concerned about human health, we're concerned about domestic animal health, and we're concerned about wildlife health, and they all play a role in the transmission of this virus, and we really need to think about it, I think, from a One Health perspective. And we need to have good communications between the different stakeholders and different groups, and all, I think all that is really important, so I am really glad I'm here and having an opportunity to tell you a little bit about the work we've been doing in wildlife. The other thing I'll just mention is, with a One Health approach, I think it's really important to recognize that we need to, you know, optimize health of humans and domestic animals and the environment, including wildlife. So, you know, I've had people say to me, oh, let's just get rid of the wild birds, but I just want to put it out there that that is not going to work. There's no benefit to be gained by attempting to control this virus through culling of wild birds or habitat destruction. It doesn't work. We shouldn't be killing wild birds to try to control this virus. And in fact, high levels of protection of wetland habitat and trying to keep wild birds away from our human-dominated areas, so some of these farm environments, trying to keep them in more wild birds in more natural areas, is going to be much more beneficial. And the key to controlling this virus in the farm environments is really biosecurity. We need to make sure that we're doing a good job with our biosecurity measures. Okay, all that being said, we do need wildlife surveillance to understand the role of different wildlife hosts and the ecology of this virus. We know now that for this particular strain, wild birds can maintain the virus, can also be infected, and potentially transmit it to other species. And these other animals, including some wild birds and wild mammals, are dying of the virus. So this is really important from a wildlife health perspective, from my perspective in terms of understanding the impact on species and species at risk. But it's also essential that we understand the role of different species so that we can look at how the virus is transmitted. So from a domestic animal perspective, we need wildlife surveillance to better understand how the virus is getting onto farms, into barns, and we need to do this through surveillance at the wildlife domestic animal interface. We need to know what's going on. And so we can do surveillance and targeted research projects to try to look at that. And we currently have a project where we're working with OMAFRA, looking at wildlife use of poultry farms, and if people are interested, we could also do that. Let me know, and we can see if we can try to expand that to look at dairy farms as well if there's interest. And then from a human health perspective, we need wildlife surveillance to better understand the zoonotic risk, so the potential for transmission from wildlife or other animals to humans related to, you know, hunting or indigenous communities that are relying on you know, wild birds that may be infected as a food source. And we need to also look at the wildlife data to assess how the virus is changing over time. And the wildlife are a good source of virus to be able to investigate that. And finally, I hope I've convinced you that wildlife surveillance is an essential component to a One Health approach to dealing with this particular strain of high path AI. And so I just wanted to put this out there. If you find a dead wild animal on your farm, you can contact us. We'd be happy to help you. We can have it tested, and you would get a report with the results, and it may, hopefully that would be useful to you. So anyway, keep that in mind. And of course, these surveillance efforts are a huge number of partners. We have, so the Canadian Wildlife Health Cooperative in Ontario, the Animal Health Lab, University of Guelph, and the Government of Ontario, and then all our federal partners and the other provincial partners are essential. And that is it for me. Thank you.